And what that means is that I'm kind of an expert on all the ways that the human genome can get futzed with during your lifetime, and which of those things cause cancer and which ones don't. Okay. So technically, that means that I'm very, very skilled in the art of DNA sequencing. Okay, I can figure out the sequence of things that I didn't know what I was looking for. And I'm also pretty good, when I say I mean the people in my laboratory. You're not going to hear their names, but there's a group of people that do this excellent work. We're really good at detecting foreign pieces of DNA in places where they're not supposed to be, even if they're real low levels. And we used those skills during the pandemic. We invented the COVID test that many of you did, a spit test. Okay, that came out of my lab because we were really good at that kind of stuff. People. Right?
Okay. I'll talk to you more about that later.
Yeah, this is our deal. This is why I know this should have been done at the federal level. Okay. So we took all these pieces of DNA and we used them to glue together what the source DNA must have been. This is kind of, again, this is what we do in the lab all the time. And all these little red and green lines here, these are all independent little pieces of DNA. This must have had 100,000 pieces of DNA in this sequencing run. And you can put them all back together and see what they came from is this circle over here. It's a plasmid that you can go shopping online to buy from Agilent. And it's clear that Pfizer took this plasmid and then they cloned spike into it, and they used it in a process called in vitro transcription translation, in vitro transcription, where you feed an RNA polymerase this plasmid and it makes a whole bunch of mRNA copies for you. Okay, and then you take this mRNA, you mix it with the lipid nanoparticle transfection reagent, and now you've got your mRNA vaccine, but they failed to get the DNA out before they did this. So these little pieces, they did make some effort to chop it up, so all these little pieces of the plasmid got packaged in with the RNA. That's clear as day what happened just from the forensics of looking at the DNA sequencing. Okay, a little bit of a regulatory note here. The way you do RNA transcription, in vitro transcription reactions, you have to give it a DNA template, okay? And you can give it a DNA template that is just a synthetic piece of DNA that is only the instructions to make the RNA, and that's what was done for getting the emergency use authorization and the clinical trial. That's called process 1 if you look up that kind of stuff. They made a PCR product of just the bits that they wanted, and then they did the in vitro transcription, made a bunch of RNA of that. There was no plasmid DNA to contaminate the stuff that was used for the trial. But that making that PCR product doesn't scale the way that was necessary to vaccinate the whole world. So a cheaper way to scale up the production of this template is to clone that PCR product into this plasmid vector, put the plasmid vector into bacteria, and then you can grow up big vats of the bacteria. They make a lot of the plasmid DNA for you. Then you use that plasmid DNA as the template to drive this transcription reaction to make your RNA. And that's how the contamination ended up in the production batches, even though it was not in the stuff that was used for the authorization trials. So I know it's a little bit of nerdy science, but it has regulatory implications for you guys. We can measure the quantity of this stuff pretty easy in the lab. We're good at doing this kind of stuff. This is the same. We made a little PC. A colleague of mine at MIT made, who used to work for the Broad Institute at MIT, he made a little PCR test, and we cloned it here. This is similar to the PCR test that you all took for the spit test. Okay, same idea and same expertise behind it. And we can quantify exactly how much of this stuff is in a vaccine or any other tissue. And, I estimate that there were about 2 billion copies of the one piece that we're looking for in every dose. And if you look back at that map I showed you where it's all these the little piece that we're looking for is just that little bit right there, okay? But if you see 2 billion copies of this, there's about 200 billion of everything else. So what this means is that there's probably about 200 billion pieces of this plasmid DNA in each dose of the vaccine,